Tyrannosaurus Rex has long been treated as a solved problem. Its size, its growth, its lifespan all appeared well understood, but recent research revealed internal evidence inside T-Rex fossils that should not exist, according to everything scientists believed. Scientists have measured its skeletons, compared its bones, calculated its weight, and reconstructed its body repeatedly over more than a century of study. Museums display towering T-Rex fossils as definitive representations of the species. The assumption has been that nothing fundamentally new remains to be discovered. That assumption rested on methods no one questioned for decades. Nearly all conclusions about Tyrannosaurus Rex came from the external features of its bones. The length of the femur, the width of the skull, the shape of the teeth. From those visible features, scientists built detailed models of growth, maturity, and lifespan. T-Rex grew fast, reached adulthood early, and lived a relatively short adult life. The model became so widely accepted, it was treated as settled fact. Bones, however, are not merely structural objects. They are biological records. Inside every fossilized bone lies a microscopic history of growth, stress, and time. These internal structures form slowly, layer by layer, while the animal is alive. Unlike surface features, they cannot be inferred from a skeleton mounted in a museum. They can only be revealed through careful internal examination.
So we have a new dinosaur that's about 150 million years old from Wyoming, from the Morrison Formation, and it's a little predatory dinosaur. It's about the size of you or me.
When scientists conducted such examinations, they found something that contradicted the established narrative. A scientific paper published in recent years described unexpected internal patterns preserved inside Tyrannosaurus Rex fossils. These patterns were real, physical, and measured directly from the bone itself. They were biological signals frozen in stone, recorded while the animal was alive. The unsettling aspect of this discovery was its implication. According to everything scientists previously believed about T-Rex growth and maturity, these internal markers should not exist. Their presence directly contradicted long-standing assumptions about how quickly T-Rex grew, how long it lived, and when it reached adulthood. This discovery does not concern finding a larger T-Rex or breaking size records. It reveals that the life story written inside the bones does not match what scientists thought they were reading. Once that contradiction became clear, researchers faced a difficult possibility. The life of Tyrannosaurus Rex may have been very different from what generations of paleontologists believed. For decades, scientists believed the life of Tyrannosaurus Rex followed a simple and efficient pattern. According to the dominant model, T-Rex grew extremely fast, reached adulthood early, and lived only a short time as a fully grown animal. This idea came from the best evidence available at the time and from reasonable comparisons with other large predators, both living and extinct. The fossil record showed that juvenile T-Rex specimens looked very different from adults. Young individuals were lighter, faster, and more slender, while adults were massive and heavily built. When scientists compared these changes, the transition appeared sudden. That visual jump led to the idea of rapid growth, especially during adolescence. A slow-growing juvenile would be vulnerable in a world populated by large carnivores. This model also aligned with what scientists knew about metabolism and biology. A massive animal requires enormous amounts of food, growing quickly, reaching full size early, and maintaining that size for a shorter adult life seemed biologically efficient. Many researchers assumed that such a large predator simply could not afford decades of slow growth. Early fossil studies reinforced this thinking. Most growth estimates were based on bone size, proportions, and comparisons between smaller and larger specimens. When scientists observed significant differences between juveniles and adults, they interpreted those gaps as evidence of rapid growth rather than long, steady development. With limited tools to examine bone interiors, there was little reason to doubt this interpretation. This view became standard. It appeared in textbooks, documentaries, and museum exhibits. T-Rex was described as an animal that grew fast, matured early, and lived a relatively short adult life, often estimated at under 30 years. The model felt complete and internally consistent. That model worked until scientists looked inside the bones. To understand how this unexpected evidence emerged, it helps to reconsider what fossils actually are. A fossil is not just a stone copy of a bone. It is a biological record created slowly while the animal was alive. Every year, every change in growth, and every period of stress leaves a trace deep inside the bone itself. Bone histology is the study of bone tissue under a microscope. Scientists take a small sample from a fossilized bone, slice it into an extremely thin section, and examine its internal structure. This process reveals patterns invisible from the outside. Because it permanently alters the fossil, it is only done when researchers are confident the information gained justifies the cost. Tree rings offer a useful comparison. When you cut through a tree trunk, you can see rings that formed year by year as the tree grew. Bones work similarly, but they are far more complex. Instead of neat circles, dinosaur bones contain multiple types of growth markers that record not just age, but changes in growth speed, health, and environment. One of the most important markers is called lines of arrested growth. These are dark bands that form when growth slows or temporarily stops, often because of seasonal changes like food shortages. Each line usually represents roughly 1 year of life. By counting them, scientists can estimate how old an animal was at death. Age alone, however, does not tell the complete story. Inside certain bones, especially weight-bearing ones like the femur or tibia, there are signals that show when an animal reached full skeletal maturity. When growth is truly finished, these markers begin to cluster tightly together near the outer edge of the bone, forming a distinct pattern. This tells scientists that the animal had stopped getting larger, even if it continued living for years afterward. Two fossils might look identical on the outside, but internally, one could belong to a young individual still growing, while the other could come from a fully mature adult. Without histology, those differences are easy to miss. Fossils do not just show what an animal looked like, they record how it lived through time. When scientists began counting those internal markers carefully in Tyrannosaurus Rex bones, the results did not align with expectations. What scientists expected to see inside Tyrannosaurus Rex bones was straightforward. Under the old model, the internal record should show a short burst of rapid growth, followed by clear signs that the animal had reached adulthood early and then stopped growing. The microscopic markers should tighten quickly near the outer edge of the bone, signaling that the skeleton had finished developing. That pattern had been assumed for decades. The bones showed something different. When researchers carefully examined the internal structure of multiple T-Rex fossils, they found growth markers continuing far longer than expected. Instead of an early end to development, the bones recorded steady growth stretching deep into the animal's life. Some specimens showed signs of skeletal maturity beginning in one major bone while remaining incomplete in another. Parts of the same animal appeared adult, while other parts were still growing. Under previous assumptions, this should not occur. A giant predator like T-Rex was thought to need to finish growing quickly. Its massive size, high energy demands, and physical stress were believed to place strict limits on how long growth could continue. A long, drawn-out growth period was considered unrealistic, yet the internal evidence directly contradicted that belief. This discovery was powerful because of its consistency. The pattern was not found in a single unusual fossil or an exceptionally preserved specimen. Similar internal growth signals appeared across multiple Tyrannosaurus Rex individuals of different sizes and ages. The same story repeated itself inside the bones. Prolonged development, delayed maturity, growth continuing far later than expected. Researchers also ruled out the most obvious explanations. These were not damaged bones. They were not distorted by fossilization. The growth markers were clean, well-preserved, and clearly defined. Different research teams, using careful methods, reached the same conclusions. The evidence was not ambiguous. For decades, scientists had assumed they were reading the fossil record correctly, but the bones themselves were telling a different story, one that did not fit the established model. If this evidence is accurate, then T-Rex was living a very different life than previously imagined. Once scientists accepted what the bones were showing, the old life story of Tyrannosaurus Rex could no longer hold. The internal growth record made one thing clear. T-Rex did not grow fast and finish early. Instead of a short, explosive growth phase, the bones revealed a long period of steady development that stretched across many years. Growth slowed gradually, not suddenly, and in several specimens, it continued well beyond the point where scientists had assumed adulthood was complete. This meant the subadult stage of T-Rex life was far longer than anyone expected. For years, possibly decades, these animals were neither small juveniles nor fully mature adults. Their bones show continued outer growth while still lacking the tightly packed maturity signals that define a finished skeleton. This extended middle phase was recorded directly in the spacing and structure of growth markers inside weight-bearing bones. Age estimates had to be reconsidered. When researchers counted growth markers more carefully, some individuals previously thought to be fully adult in their 20s showed evidence of continued skeletal development much later in life. In several cases, the internal record supported lifespans extending well beyond earlier estimates. This conclusion came from physical markers preserved year by year inside the bone tissue. These changes had real consequences for how T. rex lived. A predator that grows slowly over a long period does not behave the same way as one that reaches full size quickly. Subadult T. rex individuals would have spent many years hunting while still developing strength, mass, and bite force. They were not apex predators overnight. They earned that role gradually, as their bodies caught up with their size. This also changes how scientists think about competition. If many T. rex individuals in an ecosystem were still growing, then adults and subadults likely occupied different hunting roles and faced different pressures. The bones suggest a population structured by age and development, not just size. Survival depended not only on reaching adulthood, but on enduring a long, demanding growth period. None of these conclusions come from speculation. They come directly from the internal structure of the bones themselves, repeated across multiple specimens. The fossils were not just preserving skeletons, they were preserving life histories. Once the internal growth evidence began to challenge old assumptions, scientists had to return to a familiar question with a new level of caution. Size still mattered, but not in the simple way it once had. Instead of asking which Tyrannosaurus Rex was the biggest, researchers focused on how size was achieved and what it actually meant at different stages of life. One of the most reliable ways scientists estimate body mass in large dinosaurs is by examining the thickness of the femur. The thigh bone carries most of the animal's weight, and its circumference closely reflects how much mass the skeleton was built to support. A heavier animal needs a thicker femur. This relationship is grounded in biomechanics, which is why femur measurements remain central to serious comparisons. Responsible comparisons, however, require context. A thick femur does not automatically mean a fully grown adult. The internal structure of that same bone may show whether growth was finished or still ongoing. In the past, large size was treated as a clear sign of adulthood. Histological evidence now shows that this is not always true. Some individuals were already massive while their bones still recorded active growth. This creates a problem for simple rankings. Two T. rex specimens might appear similar in size, yet represent very different life stages. One could be a mature adult that had stopped growing years earlier. The other could be a subadult still adding bone tissue, strength, and mass. Without looking inside the bone, those differences are invisible. Scientists are careful to admit the limits of what they can know. Fossils are incomplete. Bones are often distorted or damaged. Mass estimates depend on assumptions about muscle and soft tissue that are not preserved. Even femur measurements, while reliable, cannot provide exact weights. What they offer instead is a range of plausible values. This uncertainty ties directly back to the unexpected discovery. If T. rex grew for much longer than expected, then size alone becomes a misleading measure. The internal growth record shows that age, maturity, and body mass do not always align neatly. The giants we compare may not be comparable in the way we once thought. As scientists began re-examining growth patterns inside Tyrannosaurus Rex bones, an older debate returned with new force. For years, some researchers had argued that T. rex fossils might actually represent more than one species. Some skeletons were more robust, others more slender. Some skulls appeared thicker, others longer. These differences looked large enough to suggest separate species rather than simple variation. Under the old model, this idea seemed reasonable. If T. rex grew quickly and reached adulthood early, then major differences in size and shape were easier to explain as belonging to different species. But the internal growth evidence complicated that explanation in a fundamental way. Histology showed that many of these differences could exist within a single species across different stages of life. Bones that looked dramatically different on the outside sometimes told very similar growth stories on the inside. Large, heavy individuals were not always older. Smaller, lighter ones were not always juveniles. Growth could stretch across decades, creating a wide range of body forms within one population. This kind of variation is not unusual in living animals. Modern species show enormous differences in size, shape, and mass, without being split into multiple species. Adult male and female animals can look strikingly different. Individuals that grow faster or slower can appear unrelated at first glance. Without understanding growth history, it is easy to mistake variation for separation. The internal structure of T-Rex bones provided a more reliable guide than appearance alone. Growth markers recorded timing, maturity, and development in ways that external shape could not. When scientists compared these internal records across specimens, the evidence increasingly pointed toward diversity within a single species rather than multiple distinct ones. The debate is not completely settled, and researchers acknowledge this. But the growth evidence made one thing clear: you cannot define a species based only on how a fossil looks. You have to understand how it grew. The fossil record remains deeply incomplete. Fossilization is not a normal outcome of death. It is an accident, layered on top of another accident. An animal must die in the right place, be buried quickly, escape scavengers, survive geological pressure.
Oh, wow. Oh my God, look at that. Look, the scales are preserved.
And then wait tens of millions of years for erosion or excavation to expose its remains. Most living creatures never meet those conditions. Most T-Rex individuals almost certainly vanished without leaving a trace. That reality changes how discoveries like this one should be understood. When scientists realize that T-Rex may have grown longer, matured later, and lived far longer than previously believed, it raises a question. If growth and lifespan were underestimated for so long, then the specimens we know may not represent the true limits of the species. The largest individual may never have been found. The oldest may never have been preserved. What we call extreme could simply be average for individuals that never entered the fossil record. Every new fossil does more than add information. It rearranges the framework itself. Long-standing assumptions are replaced, not because scientists were careless, but because the evidence was always incomplete. Bones that once seemed straightforward now carry deeper meaning when examined properly. The problem was never that the fossils were misleading. The problem was how confidently we thought we understood them. The thing that should not exist was not an error in nature. It was a gap in interpretation. The bones were always telling their story. We were not listening closely enough. Tyrannosaurus Rex is not a finished chapter. It is still an open one. Somewhere beneath layers of rock, there may be individuals that rewrite the story again. Larger, older, different in ways we have not yet imagined. Each discovery answers one question and quietly creates several more. The most dangerous assumption in science is believing there is nothing left to learn.